Good afternoon, everyone. I'm here on behalf of Will Louie, who's my recent PhD graduate from the lab, but unfortunately was not able to attend today because he's ill. But I'm going to present the data that he generated during his dissertation work. The white X means don't push?
It's the middle one.
Oh, okay, got it. So Zika cycles in forests in Africa using non-human primates and forest-dwelling species of Aedes mosquitoes primarily. And there was occasional spillover reported into people, but no widespread epidemics. Until recently, of course, in 2015 and 2016, Zika emerged and spread into Southeast Asia as well as Latin America, where a hallmark of the urban transmission cycle is that the virus amplifies in human reservoirs and uses highly anthropophilic and urban abundant mosquito species like Aedes albopictus and Aedes aegypti. So there's obviously been a lot of focus on trying to understand how and why Zika emerged when it did. So one thing that the mosquito field has focused on, and a primary finding, is that the global spread of Zika in the recent pandemic was facilitated by higher susceptibility of globally invasive Aedes aegypti mosquitoes. And this is really a meta-study where investigators took mosquitoes from different areas of the world, which are shown in the different colors, and they exposed them to increasing doses of Zika. And they found that mosquitoes from Asia and the Americas, so the areas that Zika invaded, were more susceptible to Zika infection after oral exposure, as manifest as higher oral ID50s. So one reason that Zika has spread globally is that it's very adaptable to now temperate areas as well, and this map shows places in the US where Aedes mosquitoes can inhabit. More locally, the mosquito and vector control districts consider Aedes mosquitoes invasive in California, and this map on the left shows areas that now include Aedes aegypti in the county. So you can see on the right, both of these two species have now invaded about half of the counties across the state, including areas around here, right, which are, you wouldn't typically think to be amenable to Aedes. But so when we think about successful transmission of an arbovirus by a mosquito, the virus needs to spread through the mosquito's body to get into the salivary glands to be transmitted. So how that works is the mosquito ingests a viremic blood meal. The blood meal is then taken into the midgut, which you can think of as being like the mosquito's stomach. Then the virus must successfully escape from that midgut and disseminate into the rest of the mosquito's body, where then it infects the salivary glands and is excreted into saliva so that when she refeeds, she transmits. So there's a very straightforward and standard way that we assess transmissibility of a virus by a mosquito in a laboratory setting, laboratory vector competence. And how that works is we take Zika virus, we load it into an artificial blood meal, so defibrinated sheep's blood. We warm it up to a vertebrate temperature, and then we present it to naive mosquitoes, which feed through this membrane. So then, while the mosquito is still alive, we remove the legs and the wings, and we insert the proboscis into a capillary tube with a little liquid in that, and that stimulates her to salivate and excrete virus. So then we assay the body to look at infection, the legs and wings to look at infection that has disseminated out of the midgut into the rest of the body, and then the saliva to assess transmission. And very straightforward, the way we conventionally view this is rates. So the number of individual mosquitoes that are positive divided by the total number that are exposed times 100. So the vector competence of a mosquito for an arbovirus is shaped by many different factors. There are mosquito genetic factors, there's regional differences in susceptibility of mosquitoes of the same species, and so on. And on the right side, the environmental factors, because mosquitoes live and do not thermoregulate, they're subject to the environmental temperatures. So when it's warmer, the extrinsic incubation period, so the time from ingestion of virus till capability to transmit in a mosquito, goes down with increasing temperature because the metabolic processes in the mosquito are faster when it's warmer. So all of these things, I would argue, have been fairly well studied in the context of arboviruses, with the exception really of the mosquito microbiome. And I'd argue that that's especially relative to the human field. It's a developing field that we're starting to understand more about in the context of arbovirus infections of mosquitoes. So I'm going to focus the rest of the talk on the microbiota. So Will started his project with two proposed mechanisms for how we could expect microbes would impact the vector competence of a mosquito. The first was that microbes in a mosquito prime the mosquito's innate immune system. The second is that microbes, either directly or indirectly, might actually modify the integrity of that midgut, which is the first barrier to productive infection of a mosquito by an arbovirus. And there are examples from other systems where people have characterized specific microbes and found that they can influence one or both of these two proposed mechanisms. So where does a mosquito acquire microbes? On the left, I'm showing the life cycle of a mosquito. So the eggs are laid into water or into a dry place that then floods and promotes hatching of the larvae. And so during the aqueous, larval, and pupal forms, the mosquito's living in water. And then it emerges from the pupal stage to become a terrestrial adult. So on the right here, basically a mosquito can be exposed to environmental microbes throughout its life. And prior studies by people that work in the mosquito ecology field have shown that the bacterial diversity that a mosquito encounters through its life changes over time and tends to decrease through the mosquito life stages. One other reason, besides the fact that they're adaptable to living in urban settings, that mosquitoes that both Aedes species are spreading globally, is that they like to lay their eggs in various different small volumes or filthy types of water, where you can imagine there could be lots of microbes living in those environments. However, when we do those vector competence studies that I showed you that assess transmissibility, we only ever look in clean water. That is, we take lab tap water and we grow larvae in it. So what we know is that mosquito microbiota are derived from the environment. They're initially acquired during aqueous phase. And until very recently, it was thought that the microbes are actually essential for larval development. But then a study in 2018 actually developed germ-free or axenic mosquitoes. And those mosquitoes have fitness deficits, but they still survive. So now we know that microbes are not necessarily acquired, but they're nutritionally beneficial. Many of the taxa persist throughout the life of the mosquito. And then there have been a limited number of studies that showed that selected microbial species can modify vector competence. So Will's two hypotheses when he began his project were that the microbial exposure of larval mosquitoes reduces the vector competence of adult Aedes aegypti for Zika. And his rationale for this was that microbial exposure in nature primes the innate immune system and/or enhances the development or the fitness of the mosquito, which could manifest as reduced susceptibility. And then the second question was more at the how. And he was predicting that the larval microbial exposure would actually reinforce the integrity of the midgut, limiting dissemination. The reason for this is really that the midgut serves as the first barrier to infection of a mosquito. So on his way to lab, Will would stop off at the cemetery in Davis, and collect water from grave sites as well as flower holding vessels. He would bring it into the water, or excuse me, into the lab, and he would concentrate the microbes that he found in that environment water. And then he would put that into laboratory tap water, or as a control, he wouldn't add the microbes. And then he sterilized the food that the mosquitoes received so we could eliminate alimentary source of microbes as a variable. So the first thing that he noticed when he did bacterial sequencing is that the abundance and the diversity of species in Aedes aegypti from California decreased over the course of development, which is consistent with prior literature. He also saw that the microbial communities shift during the course of development. And then the question became, how do these two different larval growth conditions condition susceptibility to Zika? So he took mosquitoes that were raised either in the lab water or the environment water, and then exposed them to artificial blood meals that he spiked either with Zika or that were not spiked at all. And then we did conventional vector competence, where his readouts were looking at Zika RNA levels assessed by qRT-PCR in the bodies, the legs and wings, and saliva. The first thing he noticed is that when he reared the mosquitoes in the environment water, they pupated faster and at higher efficiencies than larvae that were reared in the lab-derived water. And at first we thought maybe this was a feature of some chemistry of the water. And I don't have slides to show you due to time, but when he dilutes down the water, he finds that he still sees this same phenotype. So it probably has to do with the microbes. So next, his vector competence data. So mosquitoes that were reared in the water that did not have the microbes consistently were infected at higher rates and also transmitted Zika at higher rates as well. So this really shows that the presence of the microbes reduces susceptibility of Zika. And this phenomenon was true in mosquitoes of two different sources in California. So Los Angeles and Clovis, where Clovis is in the Central Valley near Fresno, so it's geographically quite close to LA, but if you look at the genetics of those two Aedes populations, they're not related, suggesting that the introductions of those populations into the state were from two different places geographically. And then he also saw that the same phenotype of reduced susceptibility in the presence of microbes was manifest with two different strains of Zika virus. He also did dose response studies and found that the ID50 of the lab water reared mosquitoes was consistently lower than the environment water reared mosquitoes. And the magnitude of the difference was quite striking, more than 100-fold. So this really confirms the hypothesis that microbial exposure of larvae reduces the susceptibility of the adult Aedes aegypti mosquitoes to Zika. So next we started trying to figure out some of the mechanisms by which this could be occurring. So he modified his design slightly where we used antibiotics to ablate the microbiome. So he took these same mosquitoes, the same environment source water from the cemeteries, and then he split the larvae that were reared in these pans into different groups, some of which received antibiotics to ablate the microbiome, both at the pupal stage and also orally in sugar water at the adult stage. And that group we're calling antibiotic pupae in adults. And then some groups did not receive antibiotics at the pupal stage, but were only administered antibiotics at the adult stage, whereas the third group didn't get antibiotics at all. Then he proceeded with the typical vector competence experiment design that I've described to you already. the first thing he did was verify that the antibiotics did in fact reduce the microbial burden in the Aedes aegypti mosquitoes, and that was in fact the case. Looking at vector competence, we saw that at both low and high Zika blood meal doses, which are shown by the two different shadings of red, that Zika virus disseminates more efficiently in these antibiotic treated mosquitoes compared to mosquitoes that were not. And the difference was most striking in the mosquitoes that received the antibiotics both during their pupal and their adult stages of their life. So next we started wondering whether this effect could be due to the presence of the microbes modifying the permeability of the mosquito midgut. So he loaded blood meals with FITSI labeled dextran as a way to measure the permeability of the midgut. And then right after the mosquitoes had ingested the blood meal, he harvested some of them to do staining to evaluate the dextran. And we also labeled the dissected midguts from a subset of the mosquitoes to stain using a flavivirus antibody to enumerate the number of Zika-infected midgut epithelial cells. So on the left here, what I'm showing is that antibiotic treatment tended to disrupt the regularity of the midgut cells in the gut epithelium. And then on the right, this is the dextran labeling where we quantified the relative amount of green inside versus outside of the mosquito midgut as a way to look at its integrity. And what he saw was that antibiotic treatment of both pupae and adults significantly increased the permeability of the Aedes aegypti midgut. We also found that the antibiotic-treated pupae and adults had a reduced density of midgut cells in the same areas that were counted across the three different groups. And also that when we looked at the magnitude of Zika staining in individual midguts, antibiotic treatment, especially of pupae and adults, resulted in more midgut epithelial cells infected in individual mosquitoes. Next, we were wondering whether these microbes could be actually modifying the way that the blood meal is digested. And that's been shown in, for example, other systems like with malaria and Anopheles mosquitoes. So with these same three groups, he took and exposed to blood, and here he actually took a commercially available hemoglobin assay that's rated for human use and adapted it to measure the amount of blood in these individual mosquitoes. So he first did some validation of this assay to show that if you homogenize the just fed mosquito, you can detect hemoglobin reliably, and also that you can take these homogenized mosquito samples out of the insectary to do the hemoglobin assay on the BSL-2 benchtop. And what he saw was that, well, so first he took mosquitoes that were just fed, and that was to control for the volume of the blood meal that the mosquitoes ingested, because you might predict that, you remember I told you the mosquitoes that grow up without the microbes tend to pupate more slowly, so you might think that they also would be smaller. So we didn't actually measure the size of the mosquitoes here. In hindsight, we totally should have done that. But we used the volume of blood that they took in as a proxy, or the volume of hemoglobin that they had as a proxy for the amount of blood that they digested, to make sure that it wasn't that there were differences in the amount of ingested blood across groups. But then the reason that he selected days 1, 2, and 3 after the feed is that is the period during which time the mosquito is digesting its blood meal. So looking first, so I should orient you, this is the hemoglobin magnitude, and each dot is an individual mosquito, where more hemoglobin means less efficient digestion. So on day 0, although there was a lot of intra-group variability in the magnitude of hemoglobin, there was not a significant difference in the mean across the groups. So this tells us that the mosquitoes ingested the same volumes of blood meals. But what was striking here was that as the mosquito is digesting the blood meal, consistently the antibiotic-treated groups show less efficient blood meal digestion. And this was true whether or not there was Zika virus in the mosquito's blood meal. In parallel, as he was doing all of these assays in the insectary, he also, thinking back to the immune priming hypothesis, performed RNA sequencing on a subset of mosquitoes in each of the 3 groups, where the goal was to see whether immune response genes were differentially expressed in response to antibiotic treatment. And the axes didn't show. So apologies here. The bottom line was that there were not a significant differentially expressed genes across the antibiotic treatments. So this sort of refuted our idea that there was some immune priming that the microbes were conferring. In concert with Jeff Attardo, who's a medical entomologist in the entomology department, he helped us look at gene ontology and found, perhaps unsurprisingly, that the main hits that were different across the groups corresponded to processes that are involved in blood meal digestion. So to bring this all together, on the top here, imagine a wild mosquito developing in an environment where there are many microbes. When that mosquito is an adult and takes a Zika-infected blood meal, the blood meal digestion process is very efficient. That reduces the ability of Zika to infect midgut epithelial cells, reduces dissemination out of the midgut, and also reduces transmissibility of the virus. By contrast, when we experimentally remove the microbes from the aqueous environment that the mosquitoes are developing in by antibiotic treatment, we reduce blood digestion, and that manifests as increased Zika infection of the midgut epithelium, increased dissemination, and increased transmission. So to conclude, Will's studies found that Aedes aegypti larvae that are reared in this lab water are more efficient Zika vectors compared to larvae that are reared in water from the outdoor environments. He found that antibiotic treatment enhances Zika infection and dissemination rates, especially when the Aedes aegypti are treated both as pupae and as adults. The microbial exposure of aegypti at all the life stages reduces susceptibility to Zika by enhancing blood meal digestion. That limits infection of the midgut epithelium and then reduces dissemination of the virus. And reducing the microbes with antibiotic treatment diminishes the expression of blood protein metabolism genes in response to Zika blood feeding. So I think, like many student projects, this leads to a lot more questions than we have answers for. But let's just talk about what I think are some of the implications. So first up, this shows us that rearing mosquito larvae in water that are collected from local environments, i.e. with microbes in them, could provide a more realistic assessment of Zika vector competence. So this should encourage us to try to better model the real world when we do these kinds of studies in the lab. I still think that we should focus efforts on trying to understand the relationship between blood digestion and immunity and the presence of microbes. And then maybe a pipe dream here is if we could identify microbes that reduced susceptibility of a mosquito to an arbovirus, we could develop more environmentally sustainable vector control methods beyond what is the current standard, which is treating adult mosquitoes with insecticide-resistant drugs when we have a lot of epidemic activity. So again, if you want to see, here are QR codes to the 2 papers that Will wrote. And again, I want to thank you for hearing me instead of him. I know this is totally supposed to be a trainee-focused symposium, but he sends his regrets that he can't be here. But I want to emphasize that this is really his project, and I'm just presenting it to you all. So I also want to acknowledge the funding that was provided to support this, which was through the Pacific Southwest Center of Excellence in Vector-Borne Diseases, which is CDC funded, as well as the UC Davis Vet Med Graduate Student Support Program. I'm happy to take questions.
Another great talk. So 2 questions. Did you look for Wolbachia in these in the bacteria? Did you see any?
So we did look for it, and we did not see it. Yeah.
Okay. And then the other question, you showed the disruption of the intestinal barrier with the antibiotics. Did you look at the lab water-grown bugs to see if they had that same disruption?
We did. So we were actually, we wanted to be sure that we weren't methodologically disrupting the midgut by doing the dissections, right? And so I didn't have time to show it, and I don't know how to access all my supplemental slides, but we did some beautiful H&E looking at just grossly the midguts. And we don't see any, at a 10x magnification, we don't see any gross differences between the lab-grown and the environment-grown midguts. So if there is some kind of change, it's not viewable at that gross level.
Thanks.
Really nice talk, Lark. I'm also really fascinated by this disruption of the midgut epithelial barrier. In the axenic mosquitoes that were published already, did they look at the midgut barrier and whether there was also disruption, or is this something specific to antibiotic treatment? Like if you add back the bacteria, will it recover?
Yeah, that's a great question. I think, so not in that initial study, I don't think they look at the midgut integrity in the axenic ones, but in subsequent studies, other groups have adapted the axenic model, and what they find is that, so the more times the mosquito takes the blood meal, the leakier her gut gets, and that's also true, and the axenic mosquitoes are even leakier. So, yeah.
Yeah, okay, great, thanks.